So I'm going to take us straight into the agenda because we've got a lot of stuff to cover today. And actually, let me share the slides with you guys as well. Yep. Right. Here's the link to the slides. So if you want to hop on there and look at them yourself, then you are absolutely welcome to. And in fact, I would encourage it. So the agenda, pretty simple. Today, the first thing we're going to do is a comfort check, basically like a mini survey. Um, trying to gauge how prepared you guys all feel for the exam right now. I really want to know whether most of you guys are feeling more prepared or less prepared. And hopefully we can alleviate any concerns using this session and, using the time that we have here today. Then the kind of usual staple activity, we've got our Unit 3 outlines. These are skeletal outlines that I will take you guys through. We'll navigate them based on, lecture concepts, organized by lecture concepts. And we'll have a couple questions to test your retention on material, as well as your understanding of material for these respective outlines. So that'll be good practice as well. And then finally, the final activity for this is the class outline. So in the last alternative virtual SI session, I did leave you guys the opportunity to create an outline for yourselves, but on your own time. And I think it would be much more productive if we all tried to create an outline for one of the lecture concepts together. So that is what that will be about. And then finally, we'll have about 5 minutes at the end of the session for Q&A. So I won't be able to answer, a multitude of questions, right? Not every single question that you throw at me, especially really big ones. So if you have a really big question or a really big concept question that you want to get answered, maybe don't ask it during the Q&A, but save it for an email and send it directly to Jessica or I, and we will answer it as best as we can before you guys take the exam, so you have that information available to you. Awesome. All right. And then real quick, if you haven't been to an alternative virtual SI session before, I'm not really going to explain the rules because there's a lot, but if you haven't, please look over this slide, or if you need a refresher, please look over the slide. This gives you all the information that you need to know, and please abide by those rules so that we can all have a very smooth time with the session, and that would be much appreciated. All right, so that is there for you guys to look at if you feel like it. And with that being said, I'm going to start us off with a poll called the comfort check. And here we go. I'm going to launch the polling right now. So this is the comfort check poll in which you guys will be letting me know, how you're feeling before the exam. Hopefully, most of you guys are at least feeling a little more prepared than less prepared for the exam. That's ultimately what we look for. But if not, that's not an issue, right? That's what this review session is for. So yeah, I will give you guys about another 30 seconds or so to answer this poll, unless everyone answers before then, in which case I'll just close the poll early. Yeah, so here are the results for the poll. Looks like it's kind of like going up as we go down from 2 to 7. That's like our main range. I'm glad to see that most people are at least feeling either neutral or a little more prepared than less prepared for the exam. So you probably want to be in that 5 to 7 range at this point before the exam. If you're taking the Tuesday exam, maybe you could be in the 3 to 4 range, and that's okay. But if you're taking the Monday exam, obviously you have one less day. So you probably want to be in the 5 to 7 area. So hopefully, even if you said 2 or 3 or 4 right now, by the end of the session, maybe we'll get you a little more caught up on material, a little more confident about your comprehension, and maybe you'll be a 5 or 6 or a 7 by the end. So although I'm not going to ask this question again, so that'll just be on you to compare your previous answer to what you feel at the end of the session. All right, cool. So I'm going to stop sharing results and I'm going to close the poll. And we're going to jump straight into the main activity, which is the Unit 3 outlines. So Unit 3 outlines, right here, I've created 1, 2, one, two, 3 outlines of material that we'll be going over today. Animal history, animal development, and animal osmoregulation / excretion. The final class outline will be for animal locomotion and anatomy. So that's the one I'm giving to you guys to make together as a class. And I do have my version of this outline covered up underneath the green. So you'll have that you'll be able to reference that later if you feel like it. But ultimately, I want us to make our own outline first, and then we can look at that one later on. So without further ado, let's get straight into the first outline, which is talking about animal history. So animal history, obviously, we looked at a lot of general things about animals. What are the defining traits of animals? When we talk about defining traits, we're really talking about things like how they move, how they eat, how they breathe. Um, do they belong to specific families or phylogenetic groups? Those are the kind of things that are defining traits in animal history that help us branch out or show where specific animals branch out from each other. And ultimately, part of that branching out process is speciation, which is, over time, the divergence within a species into, multiple different branches. And you can have either punctuated speciation or gradual speciation. Punctuated often being there's, a set amount of variations in the species, and there's no real transition between these variations. They're just separate on their own. Whereas gradual, there's kind of like a range of variations that kind of go from one extreme to another, and you can see that difference mapped across the different variations. And another important factor in reading into animal history is looking at divergent and convergent evolution. So divergent evolution, we're talking about animals that are similar or maybe one species that splits in paths that go into very different forms and functions. Whereas convergent evolution, often we're talking about two very random species that are not often related, but they develop similar forms and functions because their environments require them to have similar traits. They live either in similar environments or at least they have similar adaptations to their environments. And so looking deeper into some of the more important diversity-inducing factors, we're talking about feeding types. There's fluid feeders, there's filter feeders, mass feeders, stuff like that. Basically depends on what food is actually available in your environment. Obviously, if you suck the nectar from flowers, then you're going to be a fluid feeder because you're going to need a beak that can suck up the nectar, which is a liquid. If you're a filter feeder, maybe you're like a sperm whale or something where you use like sort of a system to or sorry, not a sperm whale, a baleen whale, where you use a system to sift out the food that you want and to get out food that you don't in like a mass consumption. And then you could also have mass feeders, like us, where we have, we feed on a lot of tough meat, or sometimes it could be, tough grass and tough foliage, and that you need to break it apart either with sharper canines or sturdier, masher teeth and stuff like that. There's also different types of sensory. There's, electromagnetic sensory, pulses, altitude sensory, temperature sensory, all these things that help animals navigate their environments, even if they don't have the best of sensory capabilities in other situations or other facets. And then obviously locomotion, how animals get around, how do they, either maybe escape prey or chase down prey, get their food, stuff like that. They need to be able to move their way around their environments fairly easily, or at least comfortably. And then when we go actually into the early table that really encompasses the phylogenetic family, the monophyletic group that really is the animal kingdom, we're looking into, more early ancestors, groups of organisms that kind of define where we are and where other organisms have branched out from. And earliest, most basic sort of animals that we kind of look to are usually sponges. They don't have any symmetry, and they are, they are what we look to as far as the beginning of the animal history that we're pretty concerned with. Obviously, there's, um, cells that developed over time, but that's not really what we're looking into. We're looking into developed organisms. Then you have stenophores, which are diploblastic. They also have radial symmetry, and so um, that's a really important, showing that transition from monoblastic to diploblastic organisms. And then finally, we look at the larger group of Parahoxoa. I think sponges, stenophores, and Parahoxoa are really the major groups that you want to look at, that you might talk about on the exam. So Parahoxoa is a very big group. Obviously, it contains cnidarians, protostomes, deuterostomes, and all sorts of smaller, identifications and stuff like that. And so, but there's a lot of ways we can actually break down the tree, not just by sponges, stenophores, and Parahoxoa. We can also talk about symmetry. We have, monoblastic organisms like sponges, they're asymmetric. Typically, diploblastic organisms have radial symmetry. And then also, typically, triploblastic organisms have bilateral symmetry. But there's an important thing about that, specifically looking into triploblastic organisms that fall under bilateria. Even though the name of the triploblastic group is bilateria, you have to remember not all the organisms under that roof are actually have bilateral symmetry, and starfish are really the most prominent exception to that rule because they have radial symmetry. And that's something you can find on Dr. Wilkins' slides. So then looking into Parahoxoa, we also have two major groups there that I talked about, protostomes and deuterostomes. They're very similar. The main distinction is that during the gastrulation and cleavage process, protostomes have a development of the mouth and the anus in a different order. They usually start with the anus and then go to the mouth. Deuterostome starting with the mouth, going to the anus. It's a very small distinction that you might not think of, but it is important in talking about how different organisms develop early on, and it can be a key factor in sort of like sifting through them. And then you know when we talk about these different, layers of organisms, whether they're monoblastic, diploblastic, or triploblastic, what we're of course referring to are the endoderm, the mesoderm, and the ectoderm. Those are the three layers that we talked about when we refer to animals. The endoderm is the digestive, respiratory, layer. The mesoderm is the muscular, circulatory, skeletal layer, and the ectoderm is the external layer, skin, also nervous system, stuff like that. So covering the outside. And important thing to remember. So obviously, the asymmetric monoblastic organisms will only have an endoderm because that's the internal layer, the very most internal layer. Diploblastic organisms will have the endoderm and the ectoderm. They won't have a mesoderm. And then triploblastic organisms will have all three of those layers. So those are the important things that factor into the different classifications of monoblastic, diploblastic, and triploblastic. And then, stepping back, obviously Parahoxoa, we have to talk about what Parahoxoa means. It essentially means a group of organisms that have Hox or Parahox genes, also called homeoboxes, that are key genes for controlling development and also for identifying large deviations in genetic material between organisms. And we don't really see this in sponges and stenophores, but starting from cnidarians and going down to protostomes and deuterostomes, those guys are all part of Parahoxoa and have the Hox-Parahox genes. Then you talk about notochords. So notochords are a sort of a development that are only present in deuterostomes. So let's make that clear. Only present in deuterostomes. And they're an indication of a potential spine development. Now, an important thing about notochords is that an animal can have a notochord early on in life, but it might get rid of that notochord as it develops into a mature adult. So not all organisms maintain their notochord or develop spines, as we refer to them, chordates, spines, vertebrae, from their notochord. So that's not always the case. But the organisms that do develop spines and retain their notochords fall under the group Chordata or the chordates, and these are vertebrates that we typically think of, and non-vertebrates can be can have notochords, but then they'll get rid of their notochords after a while. And then I've been talking about sponges versus stenophores versus Parahoxoa. Another important thing, and I talked about this earlier, cnidarians are the start of Parahoxoa as a group, but they kind of fall in the same range of sponges, stenophores, because cnidarians they're still diploblastic, and they do have radial symmetry like the stenophores. So they're very similar to stenophores in that regard. But the important distinction is that because they are in the Parahoxoa group, they have Hox-Parahox genes. And so that's a really important distinction between cnidarians and stenophores. And then obviously you have the large group, Bilateria, all triploblastic, but again, not all necessarily having bilateral symmetry, even though it can be confusing. And Bilateria is completely made up of protostomes and deuterostomes, either one, and those are the big groups in Bilateria. So that is all of animal history. With that being said, we're going to move into the first question, which is the animal history question. I'll read it out to you guys real quick, and then I will open up the poll and you guys can start answering it. So scientists are studying a newly discovered species with great interest. One of the things that they notice is that the organisms of the species have notochords early in their development. Which of these statements cannot be made about the species? All right. So that is the question. Now I'm going to open up the poll, launch it, and I'll give you guys about a minute or two to answer that poll. Just think about it real quick. And while you guys are doing that, I think it's important to mention that if you're making a study sheet or a study guide that you can use while you're taking the test, the chart that has the sponges and stenophores and cnidarians and parahawks and all those different groups, that chart should definitely be on your study guide or your cheat sheet because it's really good to be able to reference it very quickly. It comes in handy a lot, and it will definitely help you out, especially if you don't necessarily remember some specific exceptions to rules or the order of development of specific traits and stuff like that. So, all right, about half of you have answered the question. I'll give you guys about another 30 seconds or so to finish it up. All right, 5 more seconds for any last ones. And no. All right, I'm closing the poll. Let me share the results with you guys. So, most of you said that the statement that says this organism is part, or the species is part of the chordates, is a statement that cannot be made, and you guys are absolutely correct. The important thing to note here is that the notochord trait falls within the group of deuterostomes. All right, so that's first and first. So deuterostomes, that statement can be made. Then deuterostomes fall under the group Bilateria, which are triploblastic. So they definitely are triploblastic. And Bilateria falls under Parahoxoa. So they are definitely Parahoxoa. They definitely have Hox parahox genes and homeoboxes. Now, again, this is a tricky thing. Notochords, you might think that they would be chordates because notochords sound like chordates and you think they're tied together. The notochord comes first, chordates come after. So someone with a notochord is not necessarily a chordate, or a species with a notochord is not necessarily a chordate, because the notochord can be dropped off and that organism might not necessarily develop into a vertebrate who is part of the chordate group. So, but good on you. Good on you guys for mostly answering the correct answer. They are chordates. So that's a good impression for the first question. All right, I'm going to stop sharing results and we're going to move on to the next outline. Very cool. All right. So after animal history, you got to talk about animal development. Specifically, what are the different kinds of things that animals have developed to best interact with their environments and get the most out of their environments? And so early on, we look at, obviously, you guys know about mitosis and meiosis and, cells splitting and stuff like that. And so the early on process is you have a couple cells, they're multiplying, they're clumping up, and you have cleavage that kind of breaks open the cells and forms, a new structure that can lead to gastrulation. which is essentially the cells sort of taking on their own shape, separating and like forming a shape which will develop into the mouth and the anus of the organism. And this is a really important process because obviously it's setting up the foundational layout for an organism and its species. And this is often also you also refer to organogenesis because you also start developing the early organs that you need in an organism there. Animals also use diffusion commonly as a way of getting, nutrients either within their body, like so within different parts of their body, transportation, or even from the external environment. Specifically, aquatic animals are the most common users of diffusion as a form of material transport and nutrient exchange between their environments and themselves, whereas other animals don't really rely on diffusion as much for that. They use diffusion more as a internal mechanism of transportation between cells. So when we look at the hierarchy of the animal, how do we break it down? First of all, you got to look big picture, animal. Then, what's the most important functioning parts of the animal? You have the entire organ system that works in tandem with each other to balance everything out and to make sure that the animal has functioning consciousness, limbs, processing, stuff like that. Then you can look individually into specific organs, what their purposes are, what their roles are. Then you have the tissue that makes up each organ. Tissue is specialized and has certain attributes for every organ so that it is, most optimized for the organ's specific purposes and uses. Then you have the cells, which are less specific, that make up the tissue. Then you have the organelles within the cell. So it's organelles, just like the name sort of suggests, they're like the organs of the cell, and they do a lot of the processing and a big system movement within the cell. Then you have the molecules that make up the organelles. But obviously, this is a biology class, so we don't really look at molecules that often, but they cannot be forgotten. They do come up on occasion because specific chemical compounds can have a great effect on animals and their processes, as we'll see in the future. So, big thing, you have this whole system in the animal, lots of moving parts, lots of different uh, roles being carried out. How do you balance everything within the animal? Well, that's where homeostasis comes into play. Homeostasis is like a general correctional program in animals that essentially helps them maintain comfortable or optimal conditions to run every process, to allow every organ to function, to keep the animal running. And so when we look at homeostasis, there's a couple things we need to look at. First of all is bioenergetics. You guys probably won't really see a question on bioenergetics in the exam. Maybe one, but it's not really a big thing to focus on. It's generally just explaining how energy is transferred throughout the organism. How do they make up for loss of energy over time, and just generally how energy is distributed throughout animals to make sure everything is running well. But what we really want to look into is thermoregulation. It's a very big thing. How do animals make sure that they are at a great temperature for whatever systems they need to run in their body, processes, whatever? Because there are, certain processes within our bodies, animal bodies, human bodies, where the processes can only run under certain conditions, often temperature conditions. And so that's where thermoregulation comes into play. So endotherms are animals that maintain their own heat, or they produce their own heat. Often we refer to that production of heat as a metabolism, or the animal's metabolism. And it essentially generates energy, breaks down specific molecules for energy, and keeps a certain temperature in the animal often for endotherms. And so they produce their own heat. Whereas ectotherms, they don't spend as much energy producing their own heat. They can use that energy in other places, but they do need to maintain a good level of heat in their bodies. So what they do is they change their behavior in their environments such that they can receive the heat that is necessary to keep themselves moving. And typically, when we're looking at ectotherms, we often refer to cold blooded animals because they don't really have that same metabolism that keeps their blood warm at all times. They can fluctuate depending on their environment, but they'll do their best to keep themselves as uh, warm or at as good of a temperature as possible. Then we're looking into how the animals, you know, look at temperature within their own bodies. You got heterotherms. Heterotherms are animals that allow their heat to fluctuate between many different levels. Often, when we talk about heterotherms, we talk about an animal whose temperature fluctuates within a short period of time, a shorter period of time. And throughout the day. And so that's what a heterotherm is. And ectotherms and heterotherms often greatly overlap, but not always, but often. And then homeotherms are animals that keep their temperature the same at all times. And so endotherms are commonly homeotherms because they're generating a consistent metabolism that will keep up their temperature. They're not always homeotherms, but they are very commonly homeotherms. And even ectotherms can be homeotherms if they, have their behavior in such a way that they get consistent heat from their environment. And, talking about metabolism and how, energy is spent within animals, you got to look at total energy spent by animals versus energy per gram of tissue. And per gram of tissue or per kilogram of tissue is always an important phrase you'll have to look out for on the exam because it can definitely change the context of a question greatly. So when we talk about energy per gram of tissue, smaller animals are generally going to generate or need a lot more energy per gram of tissue. And this is because one, they have less cells. So each cell is, responsible for more jobs or more processes within the animal to keep it going. And that is why you look at their per gram of tissue, whereas a larger animal, they have many more cells. And so per gram of tissue, each cell is really not going to do as much. There's a lot that can be distributed throughout the animal. But obviously, a larger animal is still going to consume more total energy if they're doing the same action as a smaller animal because they are big and they need a lot of energy to move and to operate. So they still do have more total energy than smaller animals. Smaller animals only have to use more energy per gram of tissue. Then we look at, breathing. Obviously, another important part of animals because animals need to secure oxygen for themselves to live. So we have many different types of environments on this planet. And because of that, there's a lot of different ways and a lot of different breathing systems and circulatory systems for the animals that are on this earth with us. So, specifically looking at aquatic animals, they deal with a countercurrent flow, which is essentially using gills to direct water as well as O2 from that water in opposite directions, using the pressure that's developed from countercurrent flow to send oxygen to all their vital organs and their muscles and stuff like that. And they tend to have a unidirectional flow. So, you're just having, one system that's rotating in and out. Then terrestrial animals, we're talking about, the respiratory system. We're looking more at alveoli rather than gills or anything like that to capture oxygen, because alveoli are like these air sacs uh, that are often found in the lungs that have a lot of surface area, which allows hemoglobin and even the alveoli itself to capture oxygen from the air a lot more easily. And so alveoli are really important for oxygen consumption in terrestrial animals. And I'm not going to jump into the specific, circuits, heart chambers, stuff like that of the various types of land animals or even, aquatic animals, because you guys should be able to refer back to Dr. Wilkins' slides for those specifics. And that's another great diagram that you should actually have on your cheat sheets and your study guides. Then for circulatory systems, specifically in terrestrial animals, we're looking at, your pulmonary right, systemic left systems. Pulmonary system is often dealing with deoxygenated blood. So it really just recycling deoxygenated blood after it's left the muscles and organs and dropped off oxygen over there, so that it can come back to the heart and get oxygenated again so it can keep making the rounds. And you have your systemic system left where there is the oxygenated blood, and that is being directed towards the muscles and the organs. So think of like the pulmonary right is deoxygenated blood going back to the heart, and systemic left is oxygenated blood going away from the heart to the muscles and to the organs. And then you also have double circulation within terrestrial animals. And that's a big distinction because that allows for a lot of different pathways where you're not using up all the same space for the moving of blood. You can have multiple different blood, pathways moving all at once. And so double circulation can be a lot more effective, although it does require more energy as well. Awesome. So that was animal history and development. And now I'm going to ask you the second question. This one is very short comparatively. So it's very simple. Respond to the statement, all heterotherms are ectotherms. Is this a true statement or is this a false statement? I will launch that poll right now. Here are the results. So it looks like an overwhelming majority of you guys said that this statement was false, and that is absolutely true. Like I said earlier, um, heterotherms tend to be ectotherms, but that's not always the case. There are some very small exceptions. I think on Dr. Wilkins' chart, the mole rat was the exception to that rule, because mole rats are endotherms, but they're also heterotherms. So this is another important exception that you should know. These trends aren't always broadly generalized and work out like that. There are often a lot of exceptions or a couple exceptions that prevent you from saying a conclusive all this or that. That's often the case with biology. Life is complex and that's just how it is. But as long as you know that there are exceptions, you guys should be good. And it looks like most of you guys do know. And for those of you who didn't know, at least now you know. So you won't make that mistake on the exam if there's a question about that. Awesome. All right. So I'm gonna stop sharing, and we're gonna go to the third outline that I made before we get into the class outline. So the third outline is about animal osmoregulation and excretion. So the big things that we're talking about as far as osmoregulation, you have to understand the relationship between different bodies of uh, of animals and even their environments where there's liquid and solutes involved. So a hyperosmotic situation is where you have more solutes than you have solvent. And often our solvent is liquid water. We tend to talk about this with water. So let's just say water for now. You have more solutes than you have water uh, in in your system compared to another body, which would have more water than solute. So obviously in osmoregulation and osmosis, osmosis is always the balance of concentration across a gradient. And if there is a ability to balance the concentration gradient, then that will happen. And so for freshwater fish are hyperosmotic to their environment. They have more solutes uh, per, per amount of water in their body than the fresh water around them. And so this is very important because they need to make up for that by having a consistent water uptake system. And so that's the difference between them and saltwater fish. They have more more intricate ways of getting water back into their system, even as they lose it to their environment consistently. Whereas saltwater fish, they are hypoosmotic to their environment, which means that they have less solute per specific amount of water compared to their surroundings. Because obviously, as we know, the ocean is very salty, has a high concentration of salt, and therefore water will want to flow into the fish to reduce the concentration gradient, which makes the ocean so salty and high in solute. So freshwater fish, they'll be drained if they don't do anything about it. A saltwater fish, they'll be bloated or expanded if they don't do anything about it. And so saltwater fish have a better ways of retaining the solute or dropping off solute from themselves so that they don't, deal with being bloated and getting a lot of water rushing into their system. And then you also can have an isoosmotic environment where the concentration gradient between two bodies is exactly the same. There's no difference, and they have the same concentration of solute within them. So, and that would essentially be like a stable system where there wouldn't be any movement of water to fix anything. And among these, obviously osmoregulation, you could regulate it or you could conform to your environment depending on where you are and depending on how much you interact with water. So osmoregulators tend to be animals who maintain their levels of water and solute so that they are not really losing or gaining anything from their environment. They are staying at a steady level most of the time. And osmoconformers kind of freely let the water flow back between themselves and their environment to change the solute concentration throughout the course of a short period of time. And fish tend to be more like osmoconformers rather than osmoregulators, just because they have that constant exposure to water, and so that's often a solution for them. But we're more like osmoregulators. We have to make sure we maintain our water levels and stuff like that. We're not immersed in water all the time, and we don't uptake water through our skin or anything like that. So then you also have to talk about waste. Waste is often dealt with using water. So But not all groups have a lot of water at their disposal for waste. And so there's three different types of waste that we can have. Ammonia, which has a high water requirement, is very toxic. It's a liquid and is commonly dispersed by aquatic animals because they're always immersed in water. Urea, there's less water needed. It's not toxic. It's at least low in toxicity, but it does require more energy than ammonia to be, processed and to be released. Then uric acid requires very little water. So that's more common for desert animals. They have a low toxicity. It's more like a solid liquid substance, and it requires a lot more energy than ammonia to be created from whatever food or whatever things you uptake, and then to be released. So urea is what we produce, but uric acid is like a desert animal thing. Ammonia is more like an aquatic animal thing, and these are also not clear cut all the time, but those are the general trends that you can know about them. And then finally, the kidney, which is very important for maintaining, how much water we have in our systems. A lot of animals have a long loop of Henle, which is tied to the kidney, uh, which helps them retain water a lot better. Because the longer the loop is, the more the water can stay within it and stay within the system and is therefore retained within the individual. A shorter loop, it doesn't stay in there as long because it can go through faster and therefore it's not as helpful. But you don't need a long loop if you have a lot of water around you, or at least exposure to water. And then ADH is a molecule that helps with water retention as well. So when it is present, it prevents a lot of water loss. And so desert animals not only often have a long loop of Henle, but they also have often a lot of ADH presence if they're in a dire or desperate situation. All right. So that was animal osmoregulation and excretion. Oops. Accidentally messed up my slides there. So now we'll do the final poll question and then we'll move into the class outline. So this question says, mankind has finally landed on a planet outside of our solar system. However, there are some complications. The environment of the new planet will cause astronauts to shrivel up from water loss without special suits. How are the planets and humans related? I'm going to open up the poll now. You guys will have about a minute to answer. I'll share the results. Looks like most of you said that the planet is hyperosmotic to humans, and that would be correct. The reasoning here is that, so obviously it tells us that humans will shrivel up from water loss, so specifically water loss, on this planet without any special suits, just normally they would. What essentially that means is water is going from the human to the planet. And if water wants to go from the human to the planet, that means that the planet would have a higher concentration of solute than the human. And that's why water is moving towards the planet to balance those concentration gradients. And if the planet has a higher concentration of solute compared to the humans, that would make it hyperosmotic, because hyperosmotic refers to a substance or a body having a higher solute concentration within water compared to another body or another substance. But good job, you guys. Most of you got that one correct. And for those of you who didn't, um, just remember to make those distinctions. Just slow down and think about what hyperosmotic and hypoosmotic mean and which way you're relating them, right? Planet to humans is very different from humans to planet. So that'll be important to look at when you're taking your test. Awesome. So the last activity I wanted to do was the class outlines. Now we are a bit short on time. I did take a little too long with the explanation. But does anyone want to get us started? How about rather than instead of doing a study guide outline, why don't we just list off important things we think we should remember about animal locomotion and anatomy? So leg type, and feet to toe ratio. Muscle system often interacts with the skeletal system. All right. So muscle system, whoop, interaction with skeletal system for movement. Mechanisms of contraction, right? That's good too. Mechanisms of contraction. Obviously, very important to understand how muscles contract. Skeletal system supports animals. All right, skeletal system supports animals and protects organs. Types of skeletal systems. All right, types of skeletal systems. Sliding filament theory, very important. Filament theory, that kind of has to do with the mechanisms of contraction. Cost of transport. This is something that I heard from Jessica. Dr. Wilkin might not have covered in one of the lectures, or specifically lecture section 1. And I don't know that she will ask questions about it on the exam, since she didn't make a video about it for session 1, but I think that'd just be something important to look into. Contraction cycle of muscles. Contraction cycle of muscles. Yeah. I'll leave that up to mechanisms of contraction and sliding filament theory as in terms of contraction cycle of muscles. Can you explain the cost of transport? Sure. Just generally right now, the cost of transport refers to the amount of energy that an organism needs to spend to move around, and the type of makeup that they need to handle that energy cost for locomotion and for movement. So that's the cost of transport. Essentially, how much energy does it cost an animal to move around the way that they do, using their locomotion, and also what do they do to make up for that cost of transport. So that's what the cost of transport is basically all about. I see types of muscle fibers. All right, so there are different types of muscle fibers. That's good too. All right, cool. These are all very good suggestions and about important things we could all look at when we're talking about animal locomotion and anatomy. If you guys have any more suggestions, feel free to keep putting them in the chat. What I will do for now is I'll reveal to you what I had for my sort of outline on animal locomotion and anatomy. Just real quick, obviously, yes, I looked at the skeletal system, the muscular system. What are the details here? And specifically in the muscular system, I did a lot to look into the various steps of contraction in the sliding filament theory. I think this is a pretty good step-by-step explanation of how the sliding filament theory works. So if you want to look at that on your own free time, I definitely encourage you do. But this is going to be here for you if you want to reference it. And yeah, and then I also had a little note down here. The cost of transport depends on the method and the size. Not only how is the animal moving, but also how is the animal, exchanging energy and stuff and nutrients and materials within its system. And also how big is the animal? Because obviously bigger animals will probably have a, larger cost, just in general. So anyways, that is essentially the end of the session. I know I'm a little at the time. I'm at 4 o'clock, so technically it's over. But I don't mind if I get one or two more questions that you guys want to ask, because it is my fault that we kind of ran out of time. So if you have one or two questions you might want to ask, I'll be happy to answer them. But if not, you guys are free to go for the weekend. I hope you guys got a lot out of this session and got a lot from these slides, and hopefully they'll help you with your studies. A reminder, Jessica has her SI session on Monday for you Tuesday session people, your Tuesday exam people, she will have midterm review at I believe 2:30, but you should probably check on the Canvas page just to get a clarification on that. So yeah, and I will upload these slides to the SI website. I will send you guys the recording after this session once it's done processing. So you guys will have all of that material and reference material available for you. All right. So yeah, other than that, I hope you guys take care. If you guys have any more questions, you feel free to stick around. And if you have a big question, feel free to email me or Jessica about it, and we will be happy to answer before the exam. Yeah, have a good weekend, you guys. Take care. Study well, and good luck on your exam. I'm sure you guys will all do great. Hey, Raphael, do you have any questions for me before you head out? Got it. Got it. Yeah, sure. There's a lot to take notes on, so I totally understand. And I will be sending out the link to the slides, so you will also have access to this later. So you don't have to worry about rushing to get these done right now. All right. Awesome. All right, Raphael, I hope you have a good weekend and good luck on your test.